what we have prepared for you today is a presentation here where I'll give some brief introduction. Then we will go over some survey results that were generated after we finalized a survey very recently. And then the Cornell portion of this team is going to show some experimental data. Then Nate and Bruno are going to go over some very interesting thoughts and basic some preliminary data and some basic thoughts about the energy budget of coal plasma. Then George from Minnesota is going to give an update on some experimental data, and then we at UC Davis are going to also present some data. And I want to encourage everybody along the way to please ask questions and intervene. We should have built into this enough time to have time for questions as we go along. And of course, at the end, we have a discussion. We have prepared some discussion points as a template, but we can certainly go beyond it. The whole purpose is to make this as relevant to you as the audience as possible. So we will discuss whatever questions you might have. And to start out, I think it's important to start by differentiating what we might be considering hot plasma and cold plasma. So while hot plasma is about heating something up beyond the gaseous phase, cold plasma is more about applying an electrical charge and ionize a gas so that it becomes extremely reactive. And it's what we are particularly interested in as we break those hydrogen and oxygen and nitrogen bonds in the atmospheric air, which is what we normally use. But there's certainly the possibility to add different gases, noble gases, more CO2. You can do different things with the gas that you are ionizing. You can create other kinds of reactive molecules, or what maybe be referred to as radicals or species. It is particularly the composition, the concentration of those, and also their stability of these reactive species over time that are of particular interest, because they have different characteristics. They are reactive, which means that they also they interfere with the surfaces that they hit. So they can be used to sterilize, they can be used as fungicides to actually kill fungal spores. They can be binding in different ways and making membranes and seed coats and other surfaces more reactive, more penetrable, so that water can get in, so that fertilizer get in. This is something that has a lot of interest. And sort of on the side, to give you an idea, this is actually something that I'm in ongoing contact with NASA, because at the International Space Station, they grow food. They want to produce food for the astronauts as they go on missions. And plasma generated fertilizer and use of cold plasma as a technology out in space is definitely something they are looking into as well. So we're working on different aspects of use of this technology. And you can, of course, take this cold plasma, you can directly treat the seeds, so project this ionized gas, highly reactive gas, directly onto seeds themselves, or you can blow it into water and create what we refer to as plasma activated water, or what you will hear today us generally referring to as PAW. So this is what it looks like. This is our system. We have the jet being inserted into the water, and within seconds, literally, if depending, of course, on the volume, depending on the quality of the water that we are using, but very, very quickly we see a drop in pH. We see a continuous increase in conductivity. We see that the solids and the salinity of the water really changes, and we see that these reactive species that I was just referring to, they start appearing, and there's a lot of dynamics there that we are in the process of characterizing. There are companies also working on this. And of course, the key here is to stabilize some of those reactive species, because some of them, like nitrate and ammonium, of course, are of huge interest as fertilizers. So how can we maximize the concentration of those? How can we make it so that we can store it over time? Very, very interesting and very, very important. And those are aspects that we are working on. This is something we initially did not think we were going to spend a lot of time on, but we've been very, very fortunate that we've sort of on the side got some more help through Nate and Bruno. Nate has taken over from Andrew, who was on an original grant proposal, and Bruno, who's at Bioag Engineering, comes with a lot of knowledge about this. So this is very, very cool. What is important for you to remember as we move forward here is so PAW, plasma activated water, is not one thing. It depends on how long we keep this jet, how long we treat it for. So you can see these treatment time, it changes. So it's not. So we have to optimize what PAW we want to use for different applications. So that's very, very, very important part of this narrative. That is kind of like a compound. We think of the water, we can treat it for 1 minute, or we can treat it for 3 minutes or 10 minutes, and it has completely different characteristics. And some of those characteristics, it can be too acidic, it can be too reactive, basically, and kill the seeds. But if we give it just enough treatment so that it has the characteristics we are interested in, and that seem to have a potential benefit, then we can help and contribute with solutions that will benefit the growers. So the big picture here, what we're really looking at, and where this fits in, is thinking of coal plasma as something that can be used to directly produce different fertilizers. So definitely there is nitrates, there's ammonium in this PAW, and we will not share a lot of data on that at this meeting, but in upcoming meetings, we hope to have more of these meetings, these sessions. We will definitely, and in our newsletters, we will also prepare information about this. So we can definitely use it as direct fertilizer. We also believe that there are ways that the PAW may be used to actually boost the rhizomicrobial community. It may also be thought of as something that as you inject fertilizer into an irrigation system, maybe the combination of using PAW and a synthetic fertilizer, maybe that increases the performance of this, because normally a lot of the fertilizer that is being produced is actually not taken advantage of by the plants. But maybe because of the higher conductivity of the PAW, maybe it penetrates more readily into the roots of the growing plants. So there's a lot here where PAW can be investigated, and we will definitely work on aspects of this. And then, of course, we are looking at seed germination, how it may be used to boost the seeds germination, the vigor of the plant, and also help them create a stronger and more resilient root system, so that when so the transplant shock, when they're brought from a greenhouse environment and into a field setting, where does this can they overcome that shock more readily and get an earlier start? That's something we are very interested in looking at. On the pest management, being diseases and pests, it's well established that it can be used as a direct fungicide in greenhouses in particular. We are also going to show today some very, very compelling data that Patrice, my postdoc, has been generating on how it is boosting plant resistance, so making the plants more resistant to spider mites and thrips. We also have data demonstrating that actually combining the PAW with soft or organically certified pesticides can have so instead of using regular water, using PAW as the carrier is can be very, very important, and it may even boost the pesticides in other ways. And then, of course, we are interested in how it affects yield, both in terms of quantity and quality. This is something we will be testing. At the moment, we are working just with early plants, but we certainly have plans to. We will be going into field plots. We will be growing plants all the way to yield, and looking at those aspects as well. So as you can see, cold plasma can fit into many aspects of sustainable crop production, and we are today gonna show you initial data, more than preliminary because they are highly replicated, most of them. So this is, and some of these data we have actually already submitted research articles on. So this is moving forward. Other things are still going, and we are still working just with the cold plasma. We will also later on share data with you when we start working with direct treatment of the direct seed treatment. And ultimately, what we are really looking at, we are doing all the experimentation. We're doing it in Cornell, we're doing it in Minnesota, we're doing it at UC Davis, and we will be moving to field plots. So we're doing a lot of experimentation of the technology. We are looking into the energy. We now, because Bruno is joining us, we have a third system, a third cold plasma system. There's one in my lab, there's one in George's lab in Minnesota, and then there is the third one coming out of Bruno's lab. And we are doing characterization of the water, which is very, very useful. So we have the diagnostics. We have all to make very detailed analysis of the characteristics of the water, and we're looking into the stability of it over time. And then we combine that with phenotyping and quality control to really optimize and look at the economics and feasibility. So that's really where we're going with this. And this is the team as it stands right now. So I'm the PI on the grants that we have submitted so far. But George is in Minnesota. He's in food science and an expert on cold plasma. And Neil and Nate are long-standing experts and well recognized for their expertise about controlled environments. We have Tom joining us as one of the key extension people here in the Central Valley, particularly well connected with all vegetables, including tomatoes. And we now have Bruno, that I just mentioned, as our ag engineer, helping with the engineering side of things. And we have received in total at the moment 4 grants that we submitted. I was the PI on all of them, and actually we got all of them. So we have about $2.4 million over several years. So this has been very. So the point I'm trying to make here is that there's interest here. People understand this, and we are able to convey the message here. So we need more connection with the growers, with the companies that are involved, to make sure that we honor all this money that has been given to us. So we are in a very, very interesting spot, and we are very much looking forward to the coming years of research on this. So with that, I want to go back to the original. I want to hand it over to, I believe, Nick and Neil, who will now share some information about a survey that we recently presented, or we recently concluded.
You got it. Thanks, Christian. So I'll present from our group. I'm Neil. I'm a professor at Cornell University. I direct our Controlled Environment Agriculture group, and I'm based in our School of Plant Sciences. And our beloved lab manager is Nick Kazmar, who organized the data, which I'll present. So we conducted a survey of growers, farm operation managers, and owners of operations that produce and use young plants. And specifically, we think of this as like a benchmark survey to see what kinds of issues that growers have with seed germination, seedlings, young plant production, and then are they aware of plasma activated water and that it has some roles in improving plant performance or improving biotic and abiotic stress tolerance, as Christian was talking about. So we also wanted to learn about what are the species that growers might have the most issues with so that we can target these for future research trials, and gain learn about grower awareness of plasma activated water. And so we'll summarize these anonymous findings into a peer-reviewed publication that we can share with the community. So hopefully that can be a resource for everyone. So we developed a survey. We worked closely with the PIs on the PAW project to make sure it could be filled out in less than 5 minutes. We sent out survey request emails to about 100 different sources, focusing on horticultural trade journals and as well as green industry associations or greenhouse state associations. We distributed both English and Spanish language versions of the survey. And we included a short background on plasma activated water at the beginning of that survey as well. We did offer an incentive, so 5 user or respondents to the survey will be randomly chosen and receive $100 Amazon gift cards. So in terms of the results, we got back just over 120 results, with over 80 actually completing the survey to the end. And it's a pretty good representation of some of the major horticultural areas, especially California, the upper Midwest, and the Northeast. And then I'll start first with some of the demographic questions that we asked folks. So we asked what growing environments they use. And that was primarily in the greenhouse, 52%, 12% in the high tunnel, 22% in outdoor container nurseries, and then indoor vertical farms and other. In terms of the production area, the largest number of respondents fit into a larger category, so more than 5 acres of production area was by 32%. Then 20% were in the 10,000 square feet up to 1 acre, which I guess is 43,600 square feet. 19% were on that small end, less than 10,000 square feet, and then 15% were in that medium size category of 1 to 5 acres. The types of crops that were produced, and these add up to more than 100% because they could choose everything that they produce. So the largest was floriculture or ornamental crops, then young plants, either plugs or liners or tissue culture. And then that was similar to vegetables for produce, vegetables for transplants, followed by herbs, other, and berries. We asked if they produced in conventional or organic facilities, and 60% were conventional, 16% were organic only. And then I thought this was quite interesting, 24% have both conventional and organic production. So presumably they would have separate production areas for those 2 different types of crops. Then we asked some questions first about issues that they might have with seed germination and initial seed performance. So 81% of respondents do use seeds in their own production. And I guess the 19% that don't, they may purchase transplants or young plants that have already been germinated. And indeed, germination is an important issue. So 73% of the time respondents get lower germination than what is listed on the seed package. And so sometimes 14% said about half of the time. And there was this smaller number that was about 5 or 6% that was most of the time. So collectively, about 90% of the respondents do get lower seed germination rates than what they would hope to get. And then we asked if they conduct seed treatments to try to solve these seed germination issues. 45% say never, 37% say sometimes, and then there were these smaller categories of about half the time, most of the time, and hours. And this was really important to us, as this is showing us if they're willing to conduct seed treatments. Sometimes it's hot water for like virus treatment of seeds. Sometimes it's fungicides and so on. These are probably folks that would be willing to adopt plasma activated water if it was in a form that was economically viable for use and they had clear instructions on it. So we asked which crops have the most germination issues, and we put together a word cloud so we could kind of see visually. So the larger the word, the more times this came up in their responses. And actually, begonia was the largest issue that people had. Lettuce and in general perennials, which there's a large diversity of crops that are used. And then peppers, tomatoes, and cucumbers, native plants, some other specific plants like Dracaena, spinach, herbs, salvia, cleome, and so on.
Neil,
And
can you explain the Ireland word there? So why is Ireland there?
yeah, great, great. Ireland. So it's not the country of Ireland. And happy late St. Patrick's Day, everyone. I'm assuming that's bells of Ireland, so a common ornamental plant. So maybe somewhere is the word bells too. But
To the left of lettuce.
oh, thank you. Yeah, thanks, Anil. So to the left of lettuce, there's bell. So do you use seedlings in production? So this would be using plugs or liners, and 74% are yes, and 26% are no. And those that don't, it could be that they are plug producers themselves, for example, but then they sell those plugs or sell those liners. How often do you have inconsistent seedling growth? So 74% were sometimes, 14% were about half of the time, and then there was a bit of most of the time in hours. So on average, inconsistent seedling growth is an issue faced by a vast majority of the respondents. And we again asked which crops have the most seedling growth issues, and begonia was a big one here again too. Vinca, the diversity of perennial crops, peppers, which I've observed that myself. That's definitely a vegetable that is harder to produce uniformly and consistently. Verbena, herbs, lettuce. And then I don't know if you can see it, but in light colors, and we'll be able to distribute the full survey results. There's a wider range of plants that weren't as big of an issue with the majority of respondents, but still an issue with individual respondents. So then we asked, this was a writing question, what are the primary causes of seedling growth issues? So seed quality, and then variety. So I'm guessing that was also like cultivar specific issues. Environmental issues, so controlling temperature and humidity. Water could encompass a lot, probably water quality as well as over or under watering, so irrigation management. Fungal pathogens, of course, are a big one that we know about, like root rot, like Pythium and Rhizoctonia and so on. Unknown, that one's always concerning for me to see. That means that there's issues, but an operation doesn't have a good handle on what's actually causing those issues. So we hope that's an opportunity to reach out with their extension personnel in their state. So then we asked about what specific treatments they might use to improve seedling growth, and adjusting fertility, biostimulants, which are a range of exogenous compounds that can improve biotic and abiotic stress. And then environmental management were the largest categories that people followed. So again, our kind of thought is that if people are actively adjusting parameters to improve seedling growth, they also might be receptive to using plasma activated water. So then we asked a few final questions, which were awareness with plasma activated water. And so we asked if the respondents were aware of potential plasma activated water benefits for horticultural crops, and 20% said yes, they had heard of plasma activated water. 76% were no, and then a small percent was not sure. And we asked if folks are currently using plasma activated water in production. 94% had said never. And then a smaller percent, about 3%, were sometimes they use it, and about 1% was about half the time. And no one was using it most of the time or hours. Um, so we're seeing that clearly not that many people know about PAW, and very few operations actually use PAW. And when they do, it's just for a fraction of their crop or sometimes. Then we asked if they were interested in learning more. And this was rewarding to see as part of the team is that indeed 77% do want to learn more. Many people gave us then their email so that we can include them on the newsletters and the information that comes out from our group. 23% were not interested. And then we also asked if folks were interested in participating in no-cost PAW trials, and that is written into, Christian mentioned the 4 different projects that involve PAW. We have on-site trials with growers as objectives into many or most of those projects. So this is really helpful for us that 54% were indeed interested in that.
But we are also hoping, Neil, that will change over time, right? Because we're going to do
right.
a survey at the end, we hope that we can come across as convincing and working with industries and whatnot to shift this more towards people being interested in actually integrating this into trials at their facilities.
Absolutely. Yep. And I think as we continue to get results out of the group and convey those, we will hope to show that change in interest, and even just the change in awareness at first too. Yep. We asked if there are specific questions regarding PAW, and these were really helpful as they showed us kind of the practical questions that industry members have in terms of implementing a new technology. And so what is the impact on water quality, pH, and EC, which we'll actually cover with some of the discussion today. What impact will it have on biological additives that are used in water treatments, such as HydroGuard? And we know that there's people that use like beneficial trichoderma, for example, to suppress root diseases. And then what equipment is needed and how much does it cost to implement? How much space does this equipment take? I think that's a big crux of it, right? How do I practically apply it in my facility? How much does it cost? And what is the type of maintenance and things like that? Someone was interested in the added nitrogen that you get in the plug stage for organic transplant production. And that is, so one of the projects of these 4 is specific to organic production and the potential. And I'll be studying the ability to add plasma activated water as an organic nitrogen source. In addition to vigor and germination, what are the benefits on pathogens? So diseases. And so this was really great information for us. So the next steps in terms of the survey itself is we're going to collaborate with Dr. Cheng-Yan Yu. She's the Bachman Endowed Chair in Horticultural Marketing at the University of Minnesota. So she's in the same department as Nate Ahlens. And she's going to help us on the more like detailed analysis, which we'll get to dive into to explore the market potential for plasma activated water. And we'll be publishing the survey results in a peer-reviewed journal, using the survey results to inform the plant species that we use in our trials that are upcoming. And then importantly, our plan is to repeat this survey in 2 years so that we can look at changes in knowledge and adoption. So as more information gets out to these sources and we conduct the outreach and extension part of our projects, we hope that there'll be greater awareness and adoption of PAW. Okay, with that, do we have any time for questions? questions, Christian? Otherwise, I can respond
I
to things
think
in
I
chat
think that
too.
you did an excellent job, Neil. This was very good. I just want to start by saying that if as we go along with the analysis, I also think we will be sharing more about this because there's a lot to learn about this survey. We have a lot of information, right? So as we get more into the analysis, we will either via newsletters or in other ways share this information with as many people as share with the audience. Any questions, anybody?
I have a question. You had a comment on the results of those who were willing to use a seed treatment and suggested that that might indicate an interest in plasma activated water. Could there be a potential that in fact those that wouldn't use seed treatments wouldn't use them because they're organic producers, and perhaps you could have a number larger than those who were willing to use a seed treatment, willing to adopt this technology?
That's right. So maybe they're not doing seed treatments because they're thinking of like conventional fungicides or something, but they're
Right.
an organic producer. So I will say, working with the horticultural marketing specialist, we're planning to do a more like multi-factor analysis so we can like link trends in that case. So if someone responded that they're an organic producer, we can see how that affected their responses to some of these questions versus others. So we will be sorting through and kind of going more in depth. Yeah.
Great. Thank you.
Any other questions? That was a great one. Great comment, Tom. Any other? If not, I think we will hand it over to Nick to give some experimental data, some exciting results with lettuce. Yeah.
Great. Can everybody see my screen okay?
Yep.
Yes.
Okay, wonderful. All right, so great. I'm going to just briefly discuss some preliminary work that we've been doing looking at the potential of PAW in hydroponic lettuce production. So just as a background, we often use lettuce as our model system because it's commercially viable as well as it has a very quick turnaround where we can go from seed to harvest in 35 days. And then we can also look at it in terms of a single experimental unit, looking at different nutritional, lighting, as well as biostimulant. So typically what we'll do is a single head of lettuce, such as Rex, which is I consider that like our model species, within a single 1 gallon bucket. And then we add in an air stone for added aeration. So just a little bit of background, the importance of pH in hydroponics. So ideal range in hydroponic production is going to be pH of 5.5 to 5.8 for optimal nutrient availability. So we often have seen this image. So this is the major micro and macronutrients and their availability within the pH range. And so periodically we have to adjust this with either an acid or a base to come into that ideal range. Typically within commercial production, that's going to be sulfuric acid. But what we've seen here is that PAW has a really low pH. The PAW that we have has a pH of 1.7 and an EC of 12. And so I was thinking, it's like, okay, so there's benefits to adding PAW to the plant, but how does that work when we try to use it to adjust our nutrient our nutrient reservoir pH? So I put together a pilot study here, and the main purpose is observe PAW in maintaining the pH of our hydroponic nutrient solutions, and then quantify exactly how much PAW is required to hit that optimal range compared to our control acid. And then because we have that really high EC, is that affecting our nutrient solution EC? And then ultimately, is there any added benefit to our plant growth? So the method was we used as stated before, our Rex butterhead lettuce, germinated in Rockwool. These were grown in 1 gallon buckets with an air stone, with a standard hydroponic fertilizer solution, 2-part, 5-12-26, and calcium nitrate. And then so what I did was I adjusted the pH to that range using either a control of a 1 molar sulfuric acid solution or PAW, which had a pH of 1.7 and an EC of 12. This was adjusted every 4 days throughout the growth cycle. And what I did was I recorded the starting pH and EC of our nutrient solutions. And then the actual volume of either PAW or acid that was added to hit that target pH, and then what that final adjusted pH was. So here's some images at harvest of our heads of lettuce of PAW versus grown acid. The roots look really good. The heads look really good. From what you can see visually, there was no negative impact. So this is our pH control over time. So basically what this is showing is this was our starting pH. Then I added the acid down to our range, and 4 days later, it kind of creeps up, add it, creeps back up, add it. So within these 1 gallon buckets, there's not a lot of buffering capacity, so the pH fluctuates a lot. But what we see between the acid and the PAW was a very similar trend. They performed very, very similarly. But the amount of PAW that was required to adjust the pH compared to the sulfuric acid is about 200 times more. I think I was adding on average about 750 microliters of sulfuric acid, whereas at some points I needed 200 milliliters of PAW. So that shows us that our PAW, while being effective to actually lower our pH, it's a very weak acid. We need a lot of it. But how does that impact the EC over time? It's very flat. We see a little bit of variability, but it's within a point, 0.1. So we're not seeing any large impact on our EC when we're adding the PAW to the nutrient solution. So then the real thing is, how does it impact our plants? So we saw a significantly higher plant height for plants grown in PAW. In terms of plant volume, we saw the plants were significantly larger, those grown with PAW added. The chlorophyll content, this is interesting, was significantly lower for the plants grown under PAW, which we can get into later, but we're going to study that more. The fresh rate, while not significant here, was close to significant, right? It was a p-value of 0.08. So there's a trend here, but I think with more replication, we could actually suss that out to a significant result. But then our leaf count, this was very interesting. We had significantly higher amount of leaves on the plants where they had PAW added, and the leaf area was significantly larger. So overall, we're seeing a pretty big impact. We're just adding that small amount of PAW to adjust the pH is actually having a large impact on our plants. And then the roots, there was no significant difference there. So it wasn't impacting the root health. So in summary, PAW is a very weak acid. So we need really, really high amount of volume to adjust our pH solution. So maybe it's not the best product to be using as a pH controller, but because 200 times more. But when we just added that small amount, our plants grew quite a bit. We saw a significantly higher height, significantly larger volume, significantly larger leaf area, leaf count, and fresh weight almost there. So there's something happening there. We're seeing some signal that needs to be sussed out. So the next steps are going to be, I don't think we're going to look at using pH as a PAW as a pH controller, but start adding it and investigate the impact of added PAW on the growth. Use this in a larger replicated experimental design, multiple lettuce varieties, just to make sure we're seeing that trend across is actually replicated in not just one variety, but we see it in multiple ones. And I'd like to look at actually buffering the PAW to several different pH levels to see what the impact would be there. And then grow lettuce also under several different fertilizer levels to see if PAW would have an impact on fertilizer use efficiency. And that's all I have. Any questions?
Yeah, I got one.
Yeah.
Hi, Nick. Eric here from the
Hey,
Netherlands.
Eric, how you doing?
Hey, good to see you, and thanks for sharing these interesting results.
Mhm.
And I think also similar to what we recently discussed, I'm curious, were you able or have you done a sample analysis of the PAW that you used? Do you know the components that were present in terms of the nitrogen oxygen compounds?
No, I have not actually, and I would like to do that. This was given to us from University of Minnesota, who had done on their end. But I think really what we need to do is we need to get the same sensors and do that same component analysis that they're doing on there to see how that changes. So that's for the next steps.
Yeah, it would be great. We've done some similar experiments in the Netherlands in the past years, also on lettuce. So I think it would be good to exchange these results.
Right.
And then the second one, why did you use sulfuric acid? Why didn't you choose nitric acid?
That's a great
Because
question. That's a great question.
containing nitrate. So to say, would it be possible that the difference you observe are caused by the lack of sulfur in the PAW treatment?
Yeah, no, so that was another thing. I had a question going into it was, should I use sulfuric acid? Should I use nitric acid or phosphoric acid? Because those are all commonly used. Ultimately, I went with sulfuric acid just because that's what I had on hand at the time and what I most commonly use. But I think that's a really great point. And looking at alternative acids for pH control would be very informative.
And we do know commercial operators often use nitric acid. It's a little bit harder from a safety standpoint, but it does add nitrogen. I don't think our plants were deficient in sulfur at all, because we do add a decent good amount of sulfur in our fertilizer that both of the sets would have gotten.
No, I don't doubt it, Neil. The other way around, because you see the improved performance of PAW would be driven by a lower amount of sulfur.
I see what you're saying. Yeah.
And I think that's one of the potential benefits, where you see that the possibility to produce PAW on site, having your nitric acid without all the associated risks. But again, let's would love to have that discussion with you going forward.
Yeah. Okay, this is George here. We actually supply the water to Nick. We do have some data on the conductivity and ORP and all that we can share. I was just curious about something. So you mentioned you use a lot of the PAW just to be able to keep the pH down. And I was just thinking about the possibility of concentrating that PAW, right? So if we make that PAW, because first, sulfuric acid is really concentrated, if there's a possibility of evaporating some of the water and concentrating the PAW, that may solve the problem. I'm not sure how it will impact the other parameters, but that could be something we could look at in the future. So you have a much more concentrated form of PAW for such applications.
So Nicholas?
Yes.
Dave Fugito here from UC Davis. Just a quick comment. With regards to your lettuce trials, a couple of how to say it. Just one thing I would offer up. Did you measure or consider measuring NDVI of the lettuce that you're producing?
So that's I would consider that, right? But I would need either a camera that has a near infrared band on it, to get that. And so typically we don't have multi-spectrum cameras.
There's a low-cost NDVI handheld unit. Looks like a little wand. I could send it to you in the chat by Trimble Navigation.
Okay.
That we're using out here. Using it primarily to evaluate it on turf grass, but it's used in agricultural crops for looking at nitrogen curves.
Okay. Yeah, that'd be very interesting.
Yeah,
I mean,
it's only like $400. So it's not going to have the resolution of, let's say, a high-end infrared camera and drones, but it does correlate well, with the actual infrared sensors, the high-end ones. So
that's very interesting. Yeah, if you could please send that to me, I'd like to look at that.
The other question I dropped in the chat is, so one of the things I know out here at UC Davis, there's been some, conferences or meetings with regards to greenhouse grown crops and the emphasis on flavor and taste of greenhouse grown crops. So within your greenhouse trials, are you looking at trying to evaluate the impact on, the quality of taste or flavor? Because I know that's a big issue with regards to tomatoes and lettuce out here,
Right.
right? People make
So
the comment is that it just doesn't taste like field grown.
right. So I mean, for PAW trials, we are not doing sensory analysis, but I have extensive experience using a sensory analysis lab here in our Department of Food Science, where we've looked at tomatoes grown under different lighting conditions, as well as melons, baby hemp, and a variety of crops. So yes, we do have extensive experience doing that, but that's just not within the scope of this project at the moment.
Thank you.
Yep.
Great. Well, so with this, we're getting some good discussion. Actually, you may see an email popping up. I'm worried that we're going to run out of time, so I just added 30 minutes on the back end to this. We can stop earlier, but great feedback here. Very cool. But with Nick sort of implying that a lot needs to be added of the PAW and the potential of comparing that with an acid, it sets us up beautifully for the next session where Nate is going to talk about the energy budget of this stuff. So Nate and Bruno, please take it away.
Excellent. Well, thank you so much. So yeah, exactly like Christian said, for the last couple of weeks, really, Bruno and I have been diving into this a little bit. So we're getting started. And to start out, I guess, we're new PIs on this project. So if you, as an individual, are wondering who we are, you're not alone there. Dr. Bruno Carcioffi and I were added to this project after the original proposal. So the research team approached Bruno and myself to fill some research gaps in the project. I myself bring a horticultural physiology background with greenhouse industry experience. Bruno hails from UC Davis's biological and ag engineering section, where he works on projects including computational modeling of biochemical reactions, as well as thermal and non-thermal processes for food safety, like plasma activated water. So not only is Bruno contributing his intellectual expertise, but as Christian already mentioned in his introduction, he also has a plasma generator of his own. So Bruno and I, we came in and we started asking some different questions around energy and fertilizer production. Our fearless leader Christian, liked what he heard, and he kind of told us, "Hey, run with this. See what you can do. See what you can come up with a little bit here." So Bruno and I reformulated our questions into specifics. These specifics that should matter more to growers. So using cold plasma technology, we are ionizing these atmospheric gases, right? We all know this. They have the potential for plant uptake. Most interesting to us is nitrogen. This abundant and endless supply of N2 that is largely in an indigestible form for plant growth in atmosphere. So what we wish to do is break down the different species of nitrogen that plasma activated water can produce. Furthermore, how does the energy load pencil out? Now, this is not a complete life cycle assessment. In fact, it is so far from it that I hate to even mention the LCA process. However, it is a cursory look at the potential research avenue from the perspective of horticulturists, not just engineers. Now, we are aware that nitrogen is an essential plant element. It is the most abundant element present in plants. Nitrogen is a critical component of chlorophyll, amino acids, numerous enzymatic processes, and so forth. Additionally, we are all aware that we can get some interesting nitrogen species from this plasma activated water. However, the first few questions that come up to mind revolve around improving the current status of nitrogen production. Number one, what forms of nitrogen species can plants use? What is the current status of nitrogen fertilizers worldwide, and how are they synthesized? And lastly, how can we make it better? What are the benefits of using a concentrated high-intensity electrical field to generate nitrogen fixation from atmosphere? And how does the energy balance pencil out in comparison to the current production methods that we are currently using? Well, the current use. As it stands, worldwide agricultural use of nitrogen is about 240 billion pounds annually. And as the human population increases, so too will this figure. When growers are looking to increase yield, their first stop is usually increasing nitrogen. It is up for debate, but nitrogen may be the single most important element to crop production. Now, it goes without saying that the host of nutrients within a plant body are in fact a holistic network, thereby making all essential plant elements equally important. However, nitrogen can drive yields unlike any other element. The shame of it is that only 40 to 50% of applied nitrogen will actually be assimilated by the crops we grow. The remainder lost to runoff, leaches deep into soil, contaminating local freshwater systems and estuaries, resulting in problems such as coastal dead zones, massive algal blooms. You guys have probably all heard of many of these nitrogen and other elemental leaching problems. But that is for a conversation on another day, really. Today, we are interested in just the production side of things. More specifically, the energy associated with said production. This leads us to the current commercial nitrogen fixation method used across the world, the Haber-Bosch process. So nitrogen, the superabundant gas, 78% of atmosphere. However, in atmosphere, nitrogen is in the form of N2, dinitrogen gas, which is not directly available for most plants to use. This is due to an incredibly stable triple covalent bond between the two nitrogen atoms. The Haber-Bosch process fixes atmospheric nitrogen and converts it into ammonia, NH3. This process revolutionized the world, really. However, it is extremely energy intensive. In the 1930s, this process consumed about 100 gigajoules per ton of ammonia. Now that number is down to about 26 gigajoules and has approached its theoretical maximum efficiency. So this process is very costly on its own. The very nature of its production has been centralized to a handful of locations because of this as well. The hydrogen source comes from natural gas. This fact limits the regions that are capable of producing nitrogen fertilizers. Outside of the production itself, the infrastructure of the generation plant and the transportation and storage of ammonia inflate costs even further, not to mention the indirect cost of greenhouse gas emissions that total around 1.4% of worldwide emissions. That's nothing to sneeze at. Another important point to address here is that the end product of the Haber-Bosch process is anhydrous ammonia. So last I checked, anhydrous ammonia is not an available form of nitrogen for plant assimilation. Plants have specific active transporters and ion channels that require inorganic forms of nitrogen as nitrate or ammonium. Anhydrous ammonia is a gas at atmospheric pressure, so it needs to be injected into soils and then allow microorganisms to convert it over to the plant available forms of nitrogen. As a result, application comes with losses. Additionally, this gas will not dissolve in water and therefore cannot be used in a nutrient solution mix, as would be used in greenhouse applications for drip emitter fertigation or hydroponics, which means it would then have to go through another conversion step. And that conversion step is the Ostwald process. The ammonia gas created by the Haber-Bosch process is successively oxidized to nitric acid, nitric oxide by a heat catalyst, and then nitrogen dioxide by pure oxygen in the presence of a platinum gauze catalyst. The nitrogen dioxide is then absorbed in water to form nitric acid. Nitric acid is then neutralized with ammonia to form ammonium nitrate, two plant available forms of nitrogen here. We made it, but after a lot of chemistry, a lot of energy, albeit some exothermic, but also a significant load of greenhouse gas emissions lost to atmosphere, these external costs, if you will. So nitric oxide is a reactive gas that plays an important role in atmospheric chemistry by influence the production and destruction of ozone and thereby the oxidizing capacity of the atmosphere. So NO also contributes by its oxidation products to the formation of acid rain. So releasing this NO is not necessarily something we want to see in our atmosphere. We'd rather recycle it and contain it and move it back to the middle of this chemical process. However, it is not easily done. Considering the potential environmental issues caused by Haber-Bosch and the energy load that we talked about, developing green technology for nitrogen fixation is going to be pivotal. To me, CEA, Controlled Environment Agriculture, means do more with less. Using a non-thermal plasma system to concentrate a high-intensity electrical field shows really promising results at small scale. But that's just it, kind of. We are comparing small-scale laboratory measurements and extrapolations to a refined, large-scale industrial process, the Haber-Bosch. So from these numbers that we're looking at here on this slide, these are supplied by UC Davis Group. I calculated about 0.07 kilowatt hours per gram of nitrate produced. Theoretical numbers here, and in practice will likely be a little higher. And this is what we are resolved to accomplish, is gathering good baseline data that tells us a better picture of the energy usage in generating nitrogen species. In comparison to that, with the Haber-Bosch nitrogen fixation, our figure came in quite low. There is an air compressor that is used that is not factored in at this point. However, a literature review, though not entirely comprehensive at this point, also revealed a very scattered picture and differing opinions on the energy use in the Haber-Bosch, but also any plasma-generated equipment. So there are studies, however, none of them appear to agree with one another, and no one seems to want to use any standardized units for comparison. It would appear that Haber-Bosch uses approximately 0.13 to 0.4 kilowatt hours to produce 1 mole of ammonia. Conversely, plasma-activated water can range from some theoretical minimum of 0.05 kilowatt hours per mole of nitrogen species, all the way up to 0.51 kilowatt hours per mole of nitrogen. Either way you look at these figures, there are a lot of promising aspects to creating nitrogen species using non-thermal plasma, especially in the energy realm right here. For starters, this technology operates on electricity. This will make plasma-activated water compatible with renewable energy sources like wind and solar. If we address the histogram we're looking at here, the green bars in the middle represent electricity demands whilst producing nitric acid from the Haber-Bosch, which is the top line there, and plasma-based technology, the next 5 bars, different sets of these. This chart is derived from a techno-economic analysis. Regardless, they show very promising figures in the potential for non-thermal plasma generation of nitric acid that eliminates the polluting processes of Haber-Bosch and the Ostwald process. Cutting out those green bars with free energy would take us a long way toward elimination of the Haber-Bosch process. Furthermore, pod generation creates nitrogen species in the correct forms. As I already mentioned, we want the water to contain nitrate and ammonium. We would like to see that ratio to balance 7:1 respectively. Regardless, I'm eager to see all of the nitrogen species that can be generated. Even if it's ammonium, I think microorganisms can be added to facilitate the redox reactions required to create plant-available forms. So in conclusion, I think though there are some studies that address plasma-activated water as an alternative to nitrogen fixation, few really consider the plant environment. Moreover, units have not been standardized and a complete life cycle assessment has not been created. Hard data needs to be generated, and that is what we are trying to do here. We are creating standardized protocols within our generation labs across the country, from Bruno's to Christian's to George's, across the labs, across the country to address energy inputs. However, the full LCA will provide more information on these external costs. We hope to understand not only the production energy associated with nitrogen fertilizers, but shipping, storage, emissions, convenience, the hazards associated with it, and so forth. There's many more that go into a full life cycle assessment. So Bruno and I hope to lead a grant in the future, actually using this data as a bedrock to perform life cycle assessment on this topic. We think this is a great launching point and would ask all of you to consider your interest in involvement from an industry perspective. So please let us know if this is the type of work you would like to see in the future, and or if you would like to get involved. we would absolutely welcome feedback from all of you when we set up experimental designs and LCA methodology. So as Christian mentioned, we are hoping to form a closer connection with industry folk like yourselves to better understand the needs in your operations so we can target these needs in future research projects. So that's what I have for you today. And hopefully as this project continues, we can continue to add to this and understand more about nitrogen species generation. So yeah, we can talk about it. And if you'd like, open to some questions. Bruno and myself have definitely been reading a lot of papers recently.
Excellent, Nate. Any questions to Nate and Bruno at this point? I'm particularly interested in hearing what vital fluid and plasma treat, what you guys have to say about this.
Yeah, Nathan, thanks for sharing for your presentation and really can relate to what you tell here. And the picture you've taken from the economic analysis of Kevin Rauwenhorst and Annemie Bogaerts. You know, we work pretty close with Annemie.
Oh, fantastic.
We have performed LCAs on our plasma systems as well. So I would be happy to engage and to share our insights and to discuss, okay, what would be next steps forward. Basic conclusion is, unless carbon emission is going to penalize real rough, there's still quite a road ahead for plasma nitrogen fixation.
Right. Well, it's a really, really unique thing, right? Because this isn't a brand new technology. Basically, it was the way we were going to produce nitrogen originally, but then Haber-Bosch came along, and all of a sudden, fossil fuels were cheap. So we put 100 years of R&D into that, and we put nothing.
I fully agree. I think that Haber-Bosch is at the end of its life cycle, and plasma. I mean, there is a plasma fertilizer plant running up in Norway on hydropower. And still there is the good news is it is possible to achieve, and I think there's a lot of joint R&D necessary to close the gap. And let's engage on that one.
Yeah, but just bring a point that theoretically we can have producing nitrogen with lower energy with plasma theoretically.
Oh,
so
definitely true.
Reactors and work on these engineering sides and also the advantage to have it spread over the area, so we'll minimize transportation and whatever. So yeah, we need to work on that. I totally agree. Thanks.
I'll engage Polo, our CTO, who's done all these calculations on our side, and let's have the discussion. Would be
amazing. Yeah.
Thanks,
excellent, Eric. Excellent, really cool. Thank you so much. So we're running a little bit behind, but this is because of all the exciting stuff here. So George, feel free to the floor is yours, okay?
All right, thank you. Hope you can all hear me. So I'm George from University of Minnesota, and I'm going to talk about some of the work we've been doing here. This is the full team. We have Nate, who joined recently, but then my lab for my group, it is Ase and Masume, who have been doing pretty much all the work here. So our role as part of this project is pretty much to try and understand the PAW that we make, specifically to the machines that we have. We know, of course, using different machines, you will get different characteristics from the PAW. So we want to understand. We have a system here in Minnesota. There's one currently 2 in UC Davis. We want to make sure we understand the PAW that is made from them. And also, as an addition, we also want to look at how PAW affect germination rate of cereal grains. I'm a cereal scientist by training, so of course, I'm interested in looking at that. So what system do we have? We have the Dynamite IT system. It's actually made by Enercon. It's a company right next to us in Wisconsin. And it's basically a blown ion system. You basically have compressed air that is blown over an electrode that results in the production of all these reactive species. Actually, this is a very old system that the company makes. They have a lot more newer systems right now. So the first question we had was, how does water change when you treat it with cold plasma at different times, and specifically to the systems that we have? So I have my trusty assay do all this process. This is the setup we have. This is our unit, and then this is how we do our treatment. So for this experiment, we took city water and deionized water because we wanted to see whether using either of them have some advantages or not. So we treated 1 liter each from these, and they were treated. We took samples and we treated them for 5, 15, 30, and 60 minutes. And these were independently treated. Okay. So this here is showing how much water we lost because we're bubbling the system in the water. It's going to be heated. You're going to lose some of the water. And we lost about 13 to 18%. I would say this loss of water will depend on the kind of container you are using for your treatment. So it is important to know that for all the experiments we did, we actually let the PAW rest for 24 hours. Why? If you look at this graph here, you could see that this is water before treatment, of course, and then after we treated it for the 60 minutes, we got, of course, an increase in conductivity and oxidative reductive potential and the salt and all that. But after 24 hours of just letting it sit, we saw an increase, which means that that water is still reactive. I mean, if you use it right after treatment, it is going to be a different water the next day if you were doing an experiment that would have to use the same water that you treated. So for this group, we usually will keep our samples stored for at least 24 hours before we use for any experiments. So for this, we actually measured ORP, pH conductivity, and we sent our samples out. We have a water research lab here where they did a slew of analysis. We did all the mineral contents, nitrate, nitrite, sulfite, chloride. You can mention, it's actually a very long list, but I cannot present all of them today. This is how our samples were prepared prior to sending it to the lab for the water analysis lab for analysis. So, and we all know this happens already. So for the system that we have here in Minnesota, from that 1 liter of water we treated, we saw a significant reduction in pH. The A, B, C, D indicates statistical significance. If it's different, it means it's significant. So we saw a pH drop from 7 for tap water to about 2.13 after an hour of treatment. The same was seen for deionized water, so it doesn't really matter. We thought whatever was in that deionized, whatever was in the tap water would have a much more significant impact. Of course, there are slight differences in the values that we got, but generally pH decreased. Conductivity significantly increased with treatment time, whether or not you use tap water or deionized water. And after an hour of treatment, our conductivity was in the 5,000s microsiemens per centimeter. Now, the conductivity for most of the water that we have in the US ranges from 200 to 800 microsiemens per centimeter. So essentially, after an hour of treatment, you are way above that. I'm saying this because somebody asked me whether they could drink PAW. I said, no, at least for now. Okay. Oxidative reductive potential is an indication of how reactive that water is. The higher the ORP, the more oxygen you have in your water. We saw an increase in oxidative reductive potential as we treated our water over time. Now, ORP is very important because it's actually a measure of how lakes or water bodies actually break down waste products. The more oxygen you have, the more you have that water, the more the ability of that water to break up organic products. So we may be also looking at using PAW for wastewater management purposes. Oxidative reductive potential is also important for the food industry. We sometimes will add oxidizers to dough, to wheat flour, just to improve the protein characteristics. Maybe we can use ORP and we can use PAW just to do that. The other side for the food industry is anything that involves fat you have to avoid PAW because the oxygen species actually break down the fat. So we are trying to figure out what application will best be suited for the use of PAW. Now, nitrates. We've been talking about nitrates. We saw a significant increase in nitrate after an hour of treatment. We're looking at from less than 0.3 milligrams all the way to about 200 milligrams after 1 hour. This is a huge, huge amount of nitrates, especially in the tap water. We saw the same in the deionized water, but we had a lot more nitrates in the tap water than in the deionized water. Nitrites was the opposite. Nitrite increased to a peak around 15 minutes. Then we saw a reduction of nitrites in the water. But we know that when we're losing this nitrite is actually maybe contributing to the amount of nitrate that we have in the water, because the nitrite is an intermediate product from the conversion of ammonia to nitrates. So this is what we found when it comes to nitrate. So as I said, we measured a lot of minerals, and this is just one or two of them that I'm presenting just to show that not only do we have an increase in nitrate and nitrite, but we have an increase in the inorganic components as well. Look at sulfur or sulfates. We saw an increase with treatment time. A similar thing was observed in the deionized water, but it was very small. So that is, we started from 7.5 all the way to 13. When you look at chlorides, it's the same trend. We started from 48 all the way to 89. So all these inorganic matter are increasing in the water, significantly contributing to that conductivity that we are seeing in the PAW. The other question we get all the time is, how long can we store PAW? Can we even store it? So we decided to look into this. And here, we also use the tap water and deionized water. We produce about 15 liters of deionized water that were treated for 60 minutes, again, using the same system. And then we store them right in the open. It wasn't any special storage conditions. We store them in the lab. So we store some of them in a fish tank. We use the fish tank to make the water, but we store them in these low-density polyethylene containers. You can get this from Cole Palmer, and we left them out just to see whether they would change. This is, and the water was actually sampled every 7 days. We went all the way to 28 days, so for about a month, roughly, just to be sure of what is happening. We could go beyond that, but a month, I think, was okay for us to do. And this is what we found, okay? So PAW treated to pH of 3.8, even after 1 month, we had it still around 3.8. So we dropped from 3.8 to 3.6. So that water is still functionalized, even after 1 month. We saw the same for the deionized water as well. It is pretty much still PAW. For conductivity, we saw a slight increase in conductivity. So that water is still reactive. It is still changing, but the changes is much more minimal as you go on with the days, except for the deionized water that significantly increased after 7 days. After that 7 days, it still stayed around 550, 580 microsiemens per centimeter. So again, that water is still functional. Oxidative reductive potential, actually, we saw a slight reduction in that reactivity of the water. And so we started from 528 to about 326 for tap water. And then for the deionized water, it kind of fluctuated, but still, even after the 28 days, we dropped from 552 to 493. So that water is still good for use. Nitrates also saw an increase over time, but it kind of stabilized again after 2 weeks. And we saw this in the tap water and also in the deionized water. What was interesting to us was the amount of nitrites. Nitrite, we were losing nitrite over time. And where did it go? And as I said, this nitrite was probably being all converted to nitrates. Now, whether it's a good thing or not, I will leave that to the expert when it comes to plant growth. But this is what I found. The other thing we also looked at is, can we use PAW to germinate cereal grains? Christian and his group are looking at horticultural crops. Well, I'm interested in cereal grains, so that's what we did. So we took the same tap water, the same PAW that we made. This time, we made it to pH 5. Why pH 5? This is because with some of the data we've collected, and especially with results from Nick, we saw that the pH 3 or 2 was actually killing plants, and pH 5 was found to be kind of the best pH for making PAW. So that's what we did. So we germinated wheat and intermediate wheat grass. This is our germination setup. You basically have this container where we have the PAW at the base. So it was filled with 500 ml of PAW. The seeds are normally left on top, and it is closed to keep all the humidity in there because you need about 90% humidity. This is an actual picture of how the seeds were arranged in it. So 50 seeds, and we actually measured 10 seeds every day to look at the length of the radicle and the plumule as the germination actually went out. And then we wash these seeds. We replace the PAW every day just to prevent any mold growth. This is what we found. So the yellow line here indicates the radical for just tap water. And this deep blue line here indicates the radical, the length of the radical for the grains that were grown with plasma activated water. And you could clearly see that big difference. The plants germinated with plasma activated water had much more longer radicals and also much more longer plumule. So the grains are actually responding to the presence of the plasma activated water. These are actual pictures of the seeds after 7 days, and you can see clearly those that are germinated. This is wheat germinated with the plasma activated water, had much more longer roots. Those roots were going for the PAW in the container, and in the PAW, they were much, much longer than in the tap water, regular tap water. This is for intermediate wheat grass, which in any case is the first perennial cereal that has been released for commercial use. It is very problematic when it comes to germination, but it responded the same. This is the plumule. This is the radical for tap water, and this is the radical for PAW, and you can see that clear difference as the days of germination goes on, and it is the same for the plumule as well. So clearly, big difference between tap water and PAW. And this is a picture. You can see that not all the seeds germinated for intermediate wheat grass. For the PAW, we see a lot more of the seeds germinating, but again, you can see how long the plumes are for those grains. So for us, this is a very good thing to see because now we can probably explore the use of PAW to stimulate the germination of this intermediate wheat grass for a lot of application, especially for malting, for beer making in general. So this is what I have to share. If there are any questions, I'm happy to answer them. And of course, happy to share that I'm from University of Minnesota. Thank you.
Excellent, George. So what I'm actually thinking is we should open, if you can unshare your screen, George.
Yeah.
What I'm wondering we should do here, I'm willing to actually sacrifice my presentation because we only have about 10 minutes left, and I wanna honor people's time. So I am thinking that we should jump straight and jump straight to the discussion here. And so if people have any questions to George, let's start there, or any comments. Okay. So what we were thinking, so what a lot of the data that I was going to show are in newsletters anyway. And I will point out that you can go to our website. You can actually just use this QR code if you want, put that on take a photo of that, and that will take you directly to the website. We have these volumes. We will in very near future have more volumes, more issues being generated. So there will be more and more information. This meeting is also being recorded, and I'm going to look at the quality and see if we can make a video out of that, and also link that here on the website. So these are sort of the questions that we thought of as that we as a group could discuss. So any of you attending, are there any of these questions that you are willing to share your thoughts or comments on?
I have a question.
Go ahead.
This is Fred from Fresh Up Greenhouses. I missed most of the presentation, but I was looking at the seed germination, the cereal seed germination, and wondering if I saw that the seedlings were taller and had more roots, but were they just as vigorous, or were they just stretched out that you might get from a growth regulator?
Yeah, so we actually, this is just preliminary data. What we're trying to do is actually grow them a little longer to see how they would grow up to. So we're actually looking at putting them in pots to see how they would grow. Is that what you were trying to ask?
Yes. Sometimes just because it's taller doesn't mean it's better.
Yeah.
Absolutely. I can, Fred, so we've done some work. So this is some of the data. You can also find that in one of the most recent newsletter. So we did see here, this is just some pepper seeds. We got some commercial seeds. You can see that we had some tomato seeds that we explicitly wanted some that had low germination. We were able to jump them from about 60% all the way up to above 80 in the case of tomato, not in a little bit of an increase in pepper. But when we also looked at the shoot length we saw in pepper, we didn't see much of an effect. But when we get to tomato, there was actually a very very strong, significant response, provided that we used what we refer to as PAW1. If we used the PAW2 or 3, it was probably too reactive. It was probably too harsh on the seeds, and then on the seedlings. So then we were getting some problems. What we're going to follow up on is we also did some imaging of these seedlings. So we're going to look at how they grow over time. So it's not just the stretching of them, but actually how they perform. So this is something we are very interested in. We can show that if we grow tomato plants, we get something like this, right? So this speaks 1,000 words, I would argue, right? So the morphometrics of these plants are much bigger. This is for reals. We also saw this in ornamentals here in a marigold. We definitely had bigger plants. We also had plants that had more flower heads. So there is definitely some value to this even after the seedling stage or the germination and seedling stage. But so let's jump back into. So are there?
Christian?
Yep.
Yeah, Christian. Dave Fugino, I had a couple of general questions, I guess, or one general question to ask to the group. So George, and maybe you were the one who stimulated it, with regards to the work that you're doing there and in the 3 different locations, is it correct to say that all PAW is not the same PAW at 3 different locations, and that you could have differences in, for example, George, you were presenting tap water versus deionized water, and that tap water, obviously, we all know could be different in Cornell and in Minnesota and in Davis. So is it, how would I say it? So how do you normalize or standardize when you're talking about these different treatments in 3 different locations and perhaps different water qualities?
Yeah, thank you.
So I can take a first stab at it, and then you can add on to it, George. So this is why we do diagnostics of the waters, right? This is why we're not that's why we are collecting all this data on conductivity and on solids and whatnot. So this is why that is absolutely key, and we are finding it surprisingly diverse and unstandardized in the published literature. So we are trying as a group to overcome that, and we are actually we believe it or not, we are shipping gallons of water across the continent here. We are sending Davis water to Minnesota, and we are receiving Minnesota water, and so we're making a 3-4 way split here where water is being sent around, and we're looking at how the same water treated with different systems, what sort of characteristics we get.
Mm-hmm. Yeah. So you're perfectly right. We're trying to understand that. So I would say, from what we have so far, whether you treat UC Davis or Minnesota water, you see that decrease in pH and an increase in conductivity, but the extent of those increases or decreases is what we are trying to understand. So for now, I would say yes, it will be different, and it also depends on the type of system you're using. Mine is an ion blown system. Some people will use DBD, some people, so we're trying to collect information on water from the 3 different sources to have a clearer picture of how it differs. And not just the 3 different sources. If someone uses agricultural water versus I use just city water from the tap, or somebody uses well water, how is it going to be different? So we are trying to understand that.
But I will say, Dave, that, I mean, your question is excellent. What is currently probably the biggest conundrum for us is how dynamic the water is after it has been treated. That the fact that it still changes weeks after it has been treated. How we stabilize that, and we are looking into ways to try to do that. And I'm very curious to hear what I saw that Eric had to step out from Vital Fluids, but we're very interested in learning more about and maybe they're not willing to share that with us, because that's part of the business here, is how do you make that water stable? Not only do you maximize the fertilization benefits of the water, but also how do you stabilize it over time in terms of chemical? Can you chemically stabilize it? What sort of containers? What is the environment? Is it how does light impact this? There are lots of practical aspects that are impacting what sort of PAW you have over space and time. So it's a very dynamic system, Dave. We are completely aware of that, but that's also exciting and an opportunity to. We are in a unique spot because we have 3 locations and we have 3 systems and we have people with very, very different skill sets being part of this team. So I think, yeah, we are very well, and the more we can get the industries involved and have them open their books about the systems and how they perform and what they have, what collaborations they have, the better. I know that Bruno is part of Bruno, maybe you can talk a little bit about the network that you are part of, where people are around the country or collecting data and looking at this from an engineering standpoint.
Yeah, we have started a collaboration with many universities across the country. And the idea is to really at least have a protocol to standardize the measurements, and then we can compare different systems, different waters, and then have at the end the goal is to have a big protocol that you don't need to follow necessarily, but you can convert one thing in another. But still, we need to add energy analysis on that because they are not covering that. So this is another point that can be very interesting in the next step, to bring those other equipments that we can have access in somehow, and then spread that and trying to understand. Going to the direction George was saying, that we need to understand how it's done, and definitively a lot of work to be done in that direction.
Excellent.
And Christian, if
yes,
you don't mind, I can interject. Just before Eric left, he did send me a couple messages in the chat, and he's like, love to talk, would love to share information with you and so forth. So it sounds like they're not going to be too close guarded over there. They're happy to open up the doors.
Oh, that's beautiful. They're good guys, and they're coming. Actually, they're sending a guy to my lab on Thursday. So he's in San Francisco, and then driving up and having a visit. So yep, absolutely. All right. Well, we are running out of time, and I wanna honor this. I think this was very good. Is there an interest? So could people maybe with a thumb or something? Are you interested in us when we feel we have enough information to so we can so it's worthy of sharing? Would you be interested in receiving another invitation and having these meetings? I'm curious to hear what people from the outside what you think about this. Would you be interested in potentially sitting in on future meetings like this?
Maybe I can hop in and answer the question for with the respect of plasma treat.
Perfect.
I have enjoyed the meeting with great information, and there is many things that are very similar here in Germany as well. So regarding the pH drop and the ORP and all that values, and I'm also happy to share data on that. So I will. I'm pretty sure to continue.
I can tell you, Thomas, that we are very pleased with our plasma treat system. It's working day and night. We're burning a lot of we're making a lot of PAW with it. It's working great, but we would certainly like to get you more involved. So we're so glad you're here and would love to. So Nate, maybe that you can maybe Thomas, you can share your email or so. Or Nate, maybe you can reach out and or put yours in so that Thomas can reach you and Bruno directly.
Yeah, it would be great.
I just put my email in the chat for you, Thomas.
Yeah, thanks.
And Christian, I would say this is really good. It really went very, very well. And next time I probably would think about getting hold of the company that made my unit,
Yes,
to join these meetings.
Right.
There's also another company called Plasma Etch. I have their system in my lab as well. I think this is the beginning of building up a consortium when it comes to all
Right.
research around cold plasma. So this is really good.
And Tom, I think is Tom still on here? Yeah. Where's Tom?
Yeah.
He's
I'm
still there. Excellent. So I think Tom, we also need to start talking more with the big growers, and Zach, maybe you can help out also get us more so that we can get them on board so they can hear firsthand what we are offering here and yeah. So this should just be open to everybody. I feel. It was intended as an advisory board meeting. That was sort of a short side on my side. I think we should just make it open to everybody.
Yeah, I think so.
But we will only do this when we feel we have enough. We will not waste anybody's time. So we're not going to already say we're going to have the next meeting on May 5th or something. We're not going to do that. We will only do it when we feel we have enough to share. But I hope you, Tom, what do you think? Was this useful?
Yeah, absolutely. I substantially increased my understanding of this process, and so yeah, and to hear from the others involved in the project, absolutely useful.
Zach, what do you think? What do you think, Zach, about the tomato growers? What do you think? Are you still there?
Yeah, sorry, just unmuting myself and getting on the screen here. Yeah, at this point in the process, helpful for me and Tom to be involved. Tomato growers are going to be interested, to get down to that level. Producer going to be interested in much more practical information.
Yep.
So,
Yep.
as you say, keep working. We'll reach out to them together when we've got something
right.
that's ready for them to see in the field. Is where I would put it. I did see someone in the Zoom. I don't know if they're still there. Robbie, that might have been Robbie Johnson.
Right.
This kind of information valuable for the greenhouse industry and those groups. So keep reaching out to them.
And we are, I mean, this team, so while both Zach and Tom and across the country or even in Germany and elsewhere, this team, I can promise you, I know I speak for everybody. We are not stuck in Davis or at our campuses. We are very applied. We are very keen to work directly with the industries. We're not just faculty here. We need your input. We need you to help us facilitate contact. We are definitely interested in getting the growers involved. So this is not yeah, we are interested in promoting. We have children, most of us, and want to see this flourish and become part of sustainable crop production and make the production of food more sustainable. So we are very, very committed on so many levels to getting this all the way out to and make this companies like Plasma Treat and Vital Fluid. We want to help them be very successful here. Everybody wins here.
And just one quick comment, highlight what Zach brought up is I think initially your target audience is definitely the transplant houses. Right. Growers could be interested that there's some technology like this out there at some point, but I think initially it's the transplant producers that are going to be your target audience.
Right, and that's very important, and that means that we are very close to what Neil and Nate are doing also in elsewhere in the country. So yeah, we are definitely targeting the greenhouse producers, even if the crop is not ultimately being produced in the greenhouse, but going out in the field later on. Yeah, so whatever Neil and Nate are doing in Minnesota and in New York is also going to be relevant here. So. Great. Well, any final comments by anybody?
I'll say good job to everyone that presented. It was really great. And
yeah.